Supplemental lecture, osmolarity and tonicity. We will investigate the plasma membrane properties in the context of structure and function, and as well remind ourselves of the fluid mosaic model, or crunchy peanut butter, with the proteins being the crunchy part and the lipid bilayer being the peanut butter. So first, we think of the components of the lipid bilayer. The phospholipid is shown here on the left. We see the aliphatic hydrocarbon lipids on the glycerol backbone. The third carbon of the glycerol backbone has a phosphate group linking it to this carbon group that has the nitrogen on it. Nitrogen has a positive charge, and the oxygen off the phosphate has the negative charge. This gives it polarity, so it is partially hydrophilic. Of course, this will be hydrophobic. Cholesterol is shown here. It also has a hydrophobic portion and a charged OH group that gives it partial hydrophilicity. So in this panel here, we're seeing how the phospholipids align to form a membrane. We see it here. This is an artificial membrane with the polar heads facing the aqueous medium on the outside and the inside of this small vesicle, as we see here, or micelle. And we see that orientation, again, forming the nonpolar hydrophobic fatty acid tail portion in the center of the membrane lipid bilayer, and the partially hydrophilic heads exposed to the aqueous medium on either side. This can be created artificially in a laboratory setting. We see a 1 millimeter opening filled with the bilayer, as we saw in that micelle in the previous slide. And then if we follow the transfer of ions from one chamber to the other, this barrier completely separates them. Then the electrodes can be used to measure the charge differential across the two, and follow the movement of charged solute particles from one chamber to the other. So that lipid bilayer membrane can be created in that artificial setting in such a way that eventually they will assemble, and light, of course, will change its reflection, and they can determine in the laboratory when it is ready to be used. Of course, that is not representative of the real cell membrane that has both the lipid bilayer portion with the cholesterol in it, but as well it has transmembrane spanning proteins of the globular type, the channel type, peripheral proteins exposed on the internal side and the external only. And then, of course, there are oligosaccharide side chains forming the glycoproteins on the outside of the membrane. So a lot of differences between the real-life membrane and the one we previously saw, but it allows for the study of the movement of molecules across the membrane. This is a high-magnification transmission electron micrograph of the edge of a cell with a nearby area. The cell exterior is shown over here. And we can very nicely see the two layers of the polar heads of the phospholipids, and then in the center of these two, the translucent or electron-lucent portion, the center, which is the hydrophobic portion. So an aqueous medium here and an aqueous medium here with the partially hydrophilic heads on either side. We see a scale bar of 100 nanometers in the lower right to give us some feel for the magnitude of the structure that we're looking at. So this is a real lipid membrane, so it will have proteins as well as the lipids. In other cases, we're going to have cells adjacent to each other, as shown here, cell green and cell blue, and their corresponding lipid bilayer membranes with the proteins and the lipids. So the interstitial fluid compartment between them might be considerably small compared to what we saw in the previous slide. So this is the intracellular compartment here, aqueous, and the intracellular compartment here in blue, aqueous, and the extracellular interstitial fluid compartment in the brownish color. So let's see where we might encounter these types of cell edges. This is a typical epithelium, in this case of the kidney. So this would be the lumen of the tubule, and then this would be the internal environment with the extracellular fluid compartment, actually the basement membrane that these cells are attached to. So aqueous, a cell membrane, aqueous, a cell membrane, and aqueous. So here's the apex or the top of this cell, the apical membrane, or the base, the bottom, the basolateral membrane, the basal membrane, excuse me. And those would be similar to that first transmission electron micrograph we saw. But here where two cells are near each other and their lateral membranes are nearly adjacent, there will be a very small amount of interstitial fluid. It is still an important component of the internal environment as some molecules try to escape between the cells, and there are, of course, tight junctions here at the apical portion of the lateral membrane. That's the zonula occludens to occlude movement from external environment to internal environment and vice versa. There is a little bit that occurs there, and we'll see that later when we study these. But basically, the zonula occludens is keeping molecules from sneaking between the cells. And this is, of course, showing selective movement of molecules either from the inside internal environment to the external, the secretion at the kidney, or absorption coming in. So these are very important processes, and we'll begin to expound on those in these next series of slides. Here we see another example of epithelium. Could very well be the kidney again, to show two pathways. The transcellular path that we just saw in the previous slide, and I had indicated that the zonula occludens does occlude movement, but not all of it. There is a paracellular path that can occur, and it's very selective. So we see the transcellular pathway can go in either direction. The basal membrane of these cells, you see, have these deep infoldings to increase the surface area for the transport. Once moved from inside the cell to this extracellular interstitial fluid compartment, of course, it'll diffuse down the concentration gradient. In the case of the kidney, it would be something that was resorbed and would diffuse into the blood and be washed away. We'll see other examples in the future of paracellular path transport. So here's a model that can be utilized in the experimental laboratory. Set up basically the same with two chambers separated by a membrane. In this case, it's frog skin between the two compartments, so we can study the type of cellular movement processes that we just looked at in the previous slide, the paracellular and transcellular pathways with an underlying connective tissue. So there's an interior or serosal side, and which has the acellular basement membrane, and the external or mucosal side that does have an epithelial cell layer. In this case, the surface of the frog skin. And again, a recording system to determine the potential across these two chambers. We'll see this again a little bit later. It's introduced now as a model of studying the cell membrane. So this is what the cell membrane actually looks like in this illustration, of course. And it has, of course, the lipid bilayer membrane and then a variety of proteins, many of which are involved in movement of molecules across the membrane. Others are involved in adhesion with the cytoskeleton on the inside and then extracellular matrix molecules on the outside. Some proteins are functioning as protein receptors. We see some oligosaccharides on the outside of this one. And as well, there might be another for recognition, which typically will have some type of glycoprotein involved as well. So there's going to be movement, diffusion across the lipid bilayer that will have different properties than those crossing through the proteins, the aqueous channels. So we can have an open channel, a gated channel that's open and closed, or an active transport for these transport transport proteins, and we'll take a look at each of those. In the upper right-hand panel, we're illustrating lipid soluble movement across the lipid bilayer, and then water-soluble movement that cannot occur because of the lipophilicity or hydrophobicity of the interior of the lipid bilayer membrane. So some of these examples are oxygen, carbon dioxide, and other small nonpolar molecules, as well as some water. So these freely pass through the membrane, the lipid bilayer portion, by simple diffusion, as we'll discuss in just a moment. And then examples here would be C6H12O6, we know as glucose, other large polar water-soluble molecules and ions such as hydrogen, sodium, potassium, calcium, chloride, along with water. So not all water freely passes through the lipid bilayer membrane. So a cell membrane has the two layers composed of mainly lipids, phospholipids, and especially those phospholipids. So this lipid bilayer is the structural foundation for the membrane. It also serves as a barrier for water-soluble molecules, as we just discussed. So in the context of the water-soluble molecules, there are diverse proteins associated with bilayer that carry out most of the membrane functions. Many of these membrane proteins are enzymes, transporters of substances across the bilayer, or receptors for extracellular substances. Other types function in cell-to-cell recognition or adhesion, as we had mentioned. So now let's take into account the concentration gradient across the lipid bilayers. So we can have a concentration gradient that is a form of energy, potential energy, that can, when the channel is open, as we see here, sodium higher outside than inside. Once the channel is open, the potential energy then becomes a kinetic energy. So the steepness of the gradient, temperature, molecular size, electrical gradients, and pressure gradients influence that rate of diffusion through the protein transport molecules. So the properties of protein-mediated movement across the cell membrane will differ in many ways from the simple diffusion across the lipid bilayer. So what is the nature of the selectivity of this membrane? It is selectively semi-permeable. So it's selective because of the proteins. This one is only for sodium, and of course it is semi-permeable because not everything can get through it. But how would a lipid-soluble molecule get through this membrane? Would pass through it freely. So in that sense, it is also not selective for that. But in the entire context, it is selective. There's the non-selective for lipid-soluble, but selectivity and specificity as a semi-permeable membrane, therefore. So when molecules are moving through these ion channels, many factors are involved. They're hydrated. So we can have, for example, lithium, sodium, potassium, rubidium, I think RB is, and cesium. And they have an ionic radius in angstroms of 0.6, 0.95, 1.33, 1.48, and 1.69. So they increase as we see going down the scale here. So this is when they are hydrated, so water's all over them, basically. And then there's a free energy of hydration in kilocalories per mole. We see is higher for the smaller molecules, and then it begins to plateau as the ionic radiance increases. So these molecules should slip through with different physical characteristics. As we can see here, we have two ways of being selective and specific in these channels. One is, let's just do selectivity for the negatively charged molecules, ions in this case. This protein has a transmembrane spanning portion that is, of course, protein and therefore amino acids. Some of the amino acids are directed toward the core of the protein where we find the channel, and they have a net positive charge exposed internally, so they are going to repel any positively charged molecule, ion, and as well it'll draw in and allow the diffusion down the concentration gradient, assuming external concentration of this blue dot higher than inside. So we have a charge component, but then also size. If this was too big, even though it'd be drawn in here, it wouldn't be able to slip through. So we've got charge selectivity and size selectivity. So let's think about the movement of molecules relative to the plasma membrane with its lipid bilayer and protein components in the sense of a concentration gradient, where in this case, each of these cases has the molecule to be moved, or solute, not to be moved, to move. This one is to be moved, is going to move with reference to high concentration outside the cell and a low concentration inside the cell. The first three examples, we see that they're moving down the concentration gradient. The final and fourth example, we see it as moving up its concentration gradient, so against the concentration gradient from low to high. We see this requires energy, and that, of course, is active transport. So let's back up for just a moment here to the first example on the left and describe and define each of these. So the first is a lipid-soluble molecule. Diffuses freely down its concentration gradient, basically no restrictions. So this is diffusion, simple diffusion, and it's the lipid-soluble substance as well as water will diffuse across. And then we see the two middle ones are passive transport, so the water-soluble substances and water diffuse through the interior of the transport proteins. No energy boost is required, and it's also called facilitated diffusion. In active transport, the specific solutes are pumped through the interior of transport proteins, requiring energy to push it from low concentration to high concentration. So again, we're thinking of these aqueous pores and molecules moving through the central portion of the pore or the ion channel. Other examples we can see would be a carrier-mediated. So this would be a simple diffusion, but through a membrane protein, so the properties would be different than the simple diffusion through the lipid bilayer. The carrier-mediated depends upon a conformational change. So when it is closed to the outside, it'll be open to the inside of the cell. The binding site for the particular molecule to be carried, these are usually quite specific, will have a high affinity, so it's going to stick to it. As soon as the ligand, the molecule, is bound to the carrier, and the carrier is ligated, it undergoes a conformational change that not only closes the inside and opens to the outside, but it decreases the affinity for this ligand and releases it, and then it will move out into the other solution. So this slide gives us an overview of this. Passive diffusion shown here in the top left. Very few physical properties restricting it. So simple passive diffusion. And then there is passive transport and, of course, active transport. So with passive transport, you have passive transport down the electrochemical gradient. We'll discuss that in a while. So you have two forces, concentration gradient and electrical charge buildup across the membrane. And then carrier-mediated passive transport. So each of these two will have different physical properties. This can be saturated, and this will not have the same linear characteristics depending upon concentration gradient as you would see for the passive diffusion. For active diffusion, we can see primary active transport on the left here, and this would be against the electrochemical gradient. So this molecule wants to come out, whatever ones are inside want to come out, and we're using energy to move more of them in. And so ATP is metabolized to ADP and PI with an energy exchange to accomplish this. Secondary active transport is against the electrochemical gradient, and it's driven by ion movement down its gradient. So two molecules will move in this case, an ion that is diffusing through the channel down its concentration gradient, and the potential energy converted to kinetic energy. Some of that kinetic energy will assist the movement of another molecule. So we'll look at some slides of the different dynamics of the rate of influx to the extracellular substrate concentration, so movement into the cell with the diffusion. In the three different examples: passive diffusion through the membrane, so it is linear all the way. Passive transport through channels reaches a point where it is slowed down because of all the biophysical properties of the membrane protein interfacing with the solute that is moving. And then carrier-mediated transport reaches saturation. So when all the carrier molecules are occupied, they reach saturation. The saturation phenomenon occurs with glucose uptake. So we can see the external glucose concentration on the x-axis and the rate of glucose uptake on the y-axis. So with passive diffusion, we see here below it is a linear relationship. The rate of uptake of glucose in this example doesn't really achieve the high levels as with the facilitated diffusion. So facilitated diffusion of glucose into cells is an extremely important mechanism. So here we see the maximum velocity, and it is never achieved through the facilitated diffusion. And the 1/2 Vmax occurs very early on, which is a constant value for this molecule, kind of acts like the Michaelis-Menten constant for an enzyme reaction. There's a more linear portion of the curve here at about the half rate. So it is possible that extracellular glucose concentrations could be so high that they cannot, the cells cannot effectively soak it up. And this is important in the kidney with glucose resorption in especially diabetes mellitus. So the extracellular, the external glucose concentration inside the renal nephron ends up in the urine because we can't bring it all in. Now, we've seen direction as a very important aspect of movement, and sometimes we have a single molecule moving and two molecules moving. So in the case of a membrane channel protein or transport protein that moves one molecule in one direction, in the direction that the molecule will move, it only moves one molecule, it will be referred to as a uniporter. In some cases, two molecules will move in the same direction, as we saw with the secondary active transport. This one going down its concentration gradient, and the potential energy converted to kinetic energy draws the other one with it. They're both going in the same direction, so it is a symporter. In the case of these, on the right-hand side, these two are coupled transport in the facilitated transport classification. If they are going in opposite directions, it is an antiporter. So it's important to establish this terminology early on. Here's an example of a synporter. A coupled transport of the facilitated transport, both molecules going in the same direction, a symporter. So in this case, we have sodium, external concentration very high, so it has a very strong gradient, and it will diffuse freely through the membrane protein under the restrictions that we saw, down its concentration gradient. So potential energy converted to kinetic energy, and an organic substrate is brought with it against its concentration gradient. So we see the substrate gradient is high inside and low outside. So this is taking advantage of the kinetic energy here, the potential converted to kinetic, to draw it in. So the co-transport with sodium renders the substrate transport against its concentration gradient energetically favorable. So we've seen potential energy being used as it's converted to kinetic energy to move a molecule. Now we can look at another way of taking potential energy in the storage form of ATP, energy input, breaking it down to have an active transport, so it's ATP dependent as well against its concentration gradient. So the active movement, then the active transport, substances move against their concentration gradient, and this requires energy in the form of ATP. In a while we'll see if this is sodium, that creates the sodium gradient to be coupled or linked to this type of co-transporter. So we'll use that combined localization of those two and events to define secondary active transport. Restricted here, we see primary active transport. So transport proteins span the lipid bilayer of the cell membranes. When they bind the solute on one side of the membrane, they undergo a reversible change in shape, and we'll look at this series of steps in a while, that shunts the solute through their interior to the other side. With passive transport, a solute simply diffuses through the protein. It's not so simple, but simpler than this. Its net movement is down its concentration gradient. With the active, the net diffusion of the solute is uphill against the gradient, and the transporting protein must be activated as by ATP energy to counter the energy inherent in that concentration gradient opposing the movement. So here we see the series of events starting on the top and rotating around in this direction to describe primary active transport. So we start at the lipid bilayer of the cell membrane with a low solute concentration inside and a high solute concentration outside. In step A, ATP donates a phosphate group to a transport protein. Phosphorylation of proteins typically creates a conformational change. We see the square conformation or rectangular shape for the ligand binding site in the non-phosphorylated form, and it changes to a circular shape showing an increased affinity for the intracellular solute. So with that conformational change in affinity, the solute now will bind with a high avidity. It likes it very much now to the transport protein. With that ligation, there's another conformational change. So the chemical fit between the protein and the solute improves on one side of the membrane when the protein is phosphorylated or energized, and the solute binds to that portion of the protein. And then conformational change. So when the solute is bound, the protein shape changes, and the bound solute ends up exposed to the fluid of the other side of the membrane. So first conformational change is close inside, open outside. Then we see a conformational change in the binding site to the rectangular shape again as we have a dephosphorylation. So dephosphorylation also creates a conformational change. So the binding site reverts to its original shape. The solute and the phosphate group are released. So we decrease affinity for the solute. The only place it can go is out. When the binding site is vacant, the protein can revert to its original shape. So a final conformational snap back to the original shape. So this is, of course, a dynamic process ongoing all the time. So ATP consumption is continuous for primary active transport. Here we see an example. Of course, the sodium-potassium ATPase pump is shown here. It is an anti-porter. So 3 sodiums will bind when it is open to the inside. The affinity for the sodium binding sites is higher in this state, non-phosphorylated. For the potassium, it is low. Once it is phosphorylated, then it changes conformation, closes inside, opens outside, changes the shape, the affinity for the sodium, so it's lower. Sodium is released, increases the affinity for the potassium. Potassium binds. Then with these unligation and ligation events, a conformational change is promoted to dump the phosphate. Phosphate is lost. We now close outside, open inside, and reset our affinities. Low affinity now for the potassium, and the potassium is released to the inside. Again, sodium is going against its concentration gradient. As well, potassium is going against its concentration gradient. And this is primary active transport and anti-Porter molecule. So this membrane pump protein, the sodium-potassium ATPase pump, kind of illustrated here, is very important. So it's the question here, is this a primary or secondary active transport mechanism? Well, it alone is a primary active transport, but as we'll see, when coupled to something like the sodium glucose symporter, then it is secondary active transport. So for secondary active transport, we need to define again the symporter and the antiporter. We already did, and we'll review that as we proceed. So an example of a secondary active transport mechanism is the sodium glucose symporter transporter. The ATP-driven sodium-potassium ATPase pump stores energy in the potential energy in the steep concentration gradient for the sodium entry into the cell. And then nearby is this molecule, the sodium glucose symport transporter. And so as the sodium diffuses back across the membrane through a membrane co-transported protein, the symporter, it escorts glucose into the cell. So I took a couple of illustrations from the previous slides to put them together in the membrane. So we've got a bunch of the sodium-potassium ATPase pumps near the sodium glucose symporters. We see that the sodium gradient outside is going to assist with the movement of glucose into the cell. Well, that covers many concepts, and we need to even look at these in greater detail. And we begin first with the passive transport of solutes, solute diffusion, and osmosis, water diffusion, if you want to think of it that way. So diffusion is the movement of solute or solvent. So osmosis is a specialized form of passive transport where we have the movement of water or solvents as well. So molecules or ions of a substance constantly collide because of their inherent energy of motion, and the collisions result in the diffusion, a net outward movement of a substance from one region into an adjoining region where it is less concentrated. In the case of osmolarity, we have the total concentration of all the solute particles in a solution. So regardless of their distribution, they have an osmolarity because they are present at a certain concentration in the solvent. Collectively, solute and solvent are a solution. So we see here the simple model. Put a little drop of red dye in water. The blue dots represent the water molecules, They're all bumping into each other, and eventually the red dots will spread out evenly throughout the solution, and now this solution will have an osmolarity greater than water because it now has something in it. We'll go through a series of illustrations to expound on all of these ideas and concepts. Again, we start in the laboratory setting. We emphasize repeatedly what's that laboratory setting and what's real-life cell membranes in the laboratory and the real-life cell membrane in a real living organism. So here we have a case of a semi-permeable membrane. Very likely it is just a porous membrane, not a lipid bilayer. So we are seeing holes in it, so this very likely is a size exclusion. So it's the only type of selectivity it would have. So in this case, we are looking at individual fluxes of this little red solute particle. There's obviously more of them in chamber 1, higher concentration, than in chamber 2, so we have a concentration gradient from chamber 1 to chamber 2. The movement, or J, the flux of these molecules will have direction from 1 to 2 and from 2 to 1. So J 2 to 1 will be occurring as well, obviously much smaller in magnitude than from 1 to 2. And over a period of time, so we have a start point, or maybe 10 seconds after a start point, and a minute after the start point before we reach equilibrium, we will have a net flux of the J1 to 2, going from left to right, chamber 1 to 2, minus that going in the opposite direction. This will be occurring at all times, but these are showing the same time point, the flux occurring instantaneously at that time. We will have a net flux of a certain magnitude. At equilibrium, there should be no net flux. So remember these static freeze frame types of things at T0, T1, Tf, the final time point when we're at equilibrium. These dynamic processes will change, and of course that's where calculus comes into evaluating an overall dynamic process. But we're going to try to keep it simple, non-calculus, but remind ourselves that these are dynamic events. We have a start point, all these time points in between, and an end point. With osmosis, we are now thinking of the movement of water. So now we have a semipermeable membrane that is selective only for water, and it is selective against the solute in chamber 2. So now water is going to be at a higher concentration here. It's all water, and at a lower concentration here. So equal volumes. So the volume has to be shared between the solute and the solvent, so there is a lower water concentration on the right. So the water is going to move down its own concentration gradient. So it's important in the context of osmosis to think of water having concentrations. So there's a net movement of water from 1 to 2, and then over time there will be equilibrium. There will be no net movement of water at all. So hydrostatic pressure, as we get more water over here with all the solute, is going to create a push to push water back, a hydrostatic pressure, and the osmotic pressure is going to be pulling the water in. Let's think of it that way. So we have these two working together. So the solute will be drawing the solvent water in. That's called oncotic pressure, and we'll see that with other studies of movement from plasma to interstitial fluid and in the kidney as well. So across capillary beds, it's very important. So at equilibrium, no net movement. It would be the same at equilibrium for following solutes, as in the first example. There should be no net movement of solutes. That's what equilibrium is. There are variants on that scheme because we know that the living organism is not just water and simple ions. So osmotic pressure is produced. In this case, we're thinking of sucrose solutions, and we'll look at various concentrations. So the percent sucrose solution is shown here, 1, 2, 4, and 6. And their osmotic pressures in atmospheres, 0.7, 1.34, 2.74, and 4.10, respectively. So there is a ratio of osmotic pressure to the percentage of sucrose, and it decreases as we increase the sucrose concentration, but it will plateau. So there's a sucrose concentration at which we're not as efficient at increasing the osmotic pressure, although the osmotic pressure will still, of course, be very high. But relative to the amount of sucrose, it's not as steep of an increase. So let's continue to consider these ideas. We have a selectively permeable membrane between two compartments, on the left and on the right. We have particles A, B, C, the little blue dots, and D. Let's not confuse these little blue circles. Here we're calling them solute. We may call them water later, but here we're thinking about it as a solute. So we have multiple solute molecules, A, B, C, and D, distributed differently on each side of this membrane. So when we think of the osmotic pressure, it equals the resistance to further net water entry. And we ask the question, what's the nature of the selectivity of these two membranes shown here? So I have superimposed and inverted a lipid bilayer with the protein, and this one is the laboratory. So it's a semi-permeable membrane, selective based upon size. This is a selective semi-permeable membrane, selective only for water-soluble molecules due to the nature of this protein. It may be size, it may be charge, all those things. Here we just have size, and we're not worried about charge. So, question, which molecule would most likely be in the dynamic equilibrium, A, B, C, or D? And we can see that particle C is distributed equally across, and these blue arrows are showing the distribution, the flux for that solute C, so it is in equilibrium. A, of course, is a higher concentration on the left, and we might expect a net flux toward the right, but it depends upon the nature of this big green one and what influence it has on that. The big green one, particle D, cannot move through, size exclusion, and as well on the left side, B, it cannot move either because of size exclusion. So it would be possible. We'd have to define all the various parameters, charge and a variety of things like that, and we'll simplify it in a while. All of these might be in equilibrium, but one thing we can see for sure is with the blue arrows indicating the movement of particle C, it for sure is in dynamic equilibrium. So passive transport, diffusion, and osmosis. First, diffusion. It's the net flux of molecules from one region to another of lower concentration via random movement. Although the diffusion in open fluid is critical to cell function, we will concentrate on diffusion through membranes and plasma membranes whenever possible. So there's a law that applies to the diffusion. It's called Fick's law of diffusion with two equations, and we see that it describes the diffusive flux of molecules across membranes. So with equation 1, we see F equals Kp A times the, in parentheses, CO minus Ci. In words, the net flux of a particle, F, equals the permeability coefficient, K sub P, I should have said before, permeability coefficient or constant. For different membranes, that can be different. But for one membrane, it's likely to be constant. The membrane surface area, A, so how much membrane are you working with? 1 millimeter is what we saw in that original illustration. And then the CO minus Cn, of course, is concentration outside minus concentration inside. So we can come up with equation 2, a proportional equation. F is proportional to A times the delta C, the change in concentration across the membrane. So in other words, net flux is proportional to the concentration difference across the membrane. That's the key thing. Net flux is directly proportional to these because they are multiplied by Kp and A. So you assume that the area is not going to change, the permeability coefficient is not going to change, so what can change are the concentrations. So the most important summary we can make from this fixed law of diffusion is that the net flux is proportional, directly proportional to the concentration difference across the membrane. So this draws our attention to that concentration difference. So we make note, simple diffusion is not a saturable process. We saw that. As the concentration difference increases, so does the rate of diffusion. Now again, here we're not thinking of diffusion through a membrane protein. This is the simple laboratory example of a selectively semipermeable membrane that is freely diffusible for the solute of interest. So Kp, K sub p, is the permeability coefficient. It reflects the ease with which a particle passes through a membrane of a certain surface area at a certain concentration. So, you know, Kp is from the formula we saw before, an algebraic manipulation to Kp on just one side. So it is a function of the type of molecule, namely its solubility in lipid, where it lies on the hydrophobic to the hydrophilic scale, especially when we're talking about the lipid bilayer for the free diffusion, the simple diffusion across it. The temperature, body temperature is typically pretty much the same. The weight of the molecule, we saw that illustrated with some ions, but it's true for all molecules. And of course, the membrane characteristics. If it is not a lipid bilayer membrane, then you're not going to have very much ease, and so the permeability coefficient would be low. If it's a lipid bilayer membrane, and we're talking about a hydrophobic or lipophilic molecule, then of course the Kp is going to be high, very high permeability coefficient. So this brings us back to this linear relationship of the outside concentration and the rate of influx. So as we increase the concentration gradient, we get an linear incremental increase in the rate of flux. And of course, if you go back to your calculus, that's a change in rate, so it gets a little complicated, but we'll leave it just as that. It's a linear relationship, and what's most important, other than the properties of the membrane and the area of the membrane, so the properties of this membrane and the area of it, what is the key factor that changes is the concentration of the particles whose movement we are following, and of course that can move through the membrane. So here we point out start T0 and equilibrium Tf, the final time point. So over time many things happen, and what we know is that at equilibrium, in this simple example, we have potassium in chamber 1 concentration equaling that of chamber 2, as well for the negatively charged, the anion, the chloride. It's going to reach this equilibrium where there's an exact concentration on each side of this membrane. Now the Donan equilibrium applies to that, but it becomes a bit more complex in a variety of ways. The first is, how do we sustain a concentration gradient for some of the molecules? Glucose is an example. Here in this illustration, we're seeing different molecules, the proteins degraded to amino acids, taken up into a cell, the complex carbohydrates to the simple sugars taken up by the cell, and the fats to the lipids. So this should be simple diffusion, as we just described. But how do we keep the concentration inside the cell lower? So we have a concentration gradient. For glucose, same thing. Glucose is a prime example. We convert glucose in glycolysis to glucose 6-phosphate, and it continues. It's a short-lived intermediary, so it's immediately converted to something else. So we sustain that concentration gradient in cells by changing the properties of the molecular structure of the molecule that comes in, so it no longer contributes to that same identical molecule's concentration gradient. Therefore, we sustain a concentration gradient of high outside to low inside. Even if we take glucose and polymerize it as glycogen, we still take it out of that picture of free glucose concentration inside the cell. This requires some energy and a variety of other things. Of course, the secondary active transport is discussed here. So metabolic reactions depend on the chemical energy inherent in concentrated amounts of molecules and ions. So cells have mechanisms for increasing and decreasing those concentrations across the plasma membrane and the internal membranes through the mechanism, as I said. So we metabolize things, change their chemical form, and we can sustain a concentration gradient. Now, this is a simple example of the Donan equilibrium concept that we talked about up above here, but the Donan equilibrium also lets us understand when additional molecules are taken into account that cannot move and what impact they have on the movement of the ones that can move. So equilibrium is going to be different in this case, and the next slide takes that at a higher magnification. So here we are adding to our potassium chloride solution a potassium anion molecule. This anion has multiple negative charges, so it's has rather strong negative net charge. So it's going to change the characteristic properties of this solution that was at equilibrium. So now what is going to happen over time when we reach equilibrium is that this negative charge is going to repel the chloride. The only thing that can move is chloride, so more of it's going to go over to chamber 2. It's going to attract the positively charged potassium, so we're going to get some potassium coming back. And so at equilibrium, potassium in chamber 1 is going to be greater than that in 2, and the chloride will be less in chamber 1 than in chamber 2. So now we're beginning to see how the Donan equilibrium changes to account for the electrical charges. So we now have an electrical chemical phenomenon occurring here. And again, it's dependent upon the nature of the membrane. It is selectively semi-permeable for molecules of this size, but not a big one like that. So for example, that big green thing we saw in the previous one, we'll see it again. So the Donan equilibrium formula can be derived by assigning values to these, Z for A, the Y plus Z for the K concentration in chamber 1, and Y for the chloride concentration. And the reason for this is that these charges have to balance out. So you have to have as many positives, Y plus Z, as you have negatives. And then you have a value of only X for the concentrations of potassium and chloride in the opposite chamber. So they should be identical. But this is not what we see inside living cells, that is what we see inside living cells relative to their interstitial fluid, such as a muscle cell here. So sodium in and out, potassium in and out, calcium in and out, chloride in and out, and an anion in but no out. So we have the anion inside, so it's going to help keep low negatively charged molecule or solute concentrations inside, 3 to 4 for chloride, versus outside, 120 millimolar. And it's going to help keep the larger ones, the negatively charged ones in, but there's things that act on calcium, so it's very low inside and higher outside. So there's more than just this anion affecting the intracellular calcium. It's very low, less than 10 to the minus 3 millimolar. And then sodium and potassium. Sodium is high inside, 140, 2.5 millimolar outside, so the anion might be impacting that. We think it would impact the sodium, but sodium is at 10 millimolar inside and 120 millimolar outside. So there must be a lot of processes going on to create this relationship of ion concentrations inside the cell versus outside the cell. Here, sodium-potassium ATPase pump pushes sodium out and brings potassium in. Sounds good. For the calcium, it is sequestered and removed as a free form diffused in the cytosol. So the free cytosolic calcium concentrations intracellularly must be kept at a very low level. If they get too high, it kills the cell. So there's a lot more happening than what we saw in our simple experimental models. So this table shows the internal and the external concentrations of some electrolytes in the specific nerve and muscle tissues. So we see the squid nerve, the crab leg nerve, and the frog sartorius muscle. And here below we see some values that we'll look at again after peeking at the next slide. So we can just take some quick looks at some of these examples. So the squid nerve tissue, 49 millimolar sodium inside, 440 out, and the ratio is 1:9. There are other examples for potassium and chloride here as well. The crab leg nerve, 52 in, 510 out, a 1:10 ratio. And the frog sartorius muscle, 10 in and 120 out, with a ratio of 1:12. So not very different ratios, but different in and out, so a lot of different environmental properties that impact it. Take a quick look at potassium, 410 inside, 22 outside, a 19:1. We look at the crab leg nerve, 410 in, 12 out, and a 34:1 ratio. So compare that to the 34:1, compare it to the 19:1, 56:1 for the frog sartorius muscle. So there's something going on with potassium in these different species. So there's the ideal laboratory situation, and then there's every living cell and every living organism and the genes in those cells that somehow a decision is made to express them differently and put them in the membrane differently. So we have different membrane properties for different cells, even identical cell types, like the nerve, peripheral nerve, for different species. So we generalize, but specifics become very important. So the Donan equilibrium would say that the potassium inside times the chloride inside should equal the potassium outside times the chloride outside if everything was ideal, but it isn't. If we look at these, for example, the potassium inside and the outside, excuse me, and the same thing for the chloride. So we end up with 16,400 not equaling 12,320. So something is impacting the dynamics of the transmembrane ion concentrations in this cell so that it is not behaving ideally based upon the Donan equilibrium. So this is true in this other cell type as well. So we know there's a lot of other things in the membrane. We've admitted that repeatedly. So now let's take a look at osmosis. Here we see, again, it is the passive movement of water from the high concentration of water, solvent, to the area where there's less of it, a lower concentration. But it will move from low solute to high solute. So the water concentration is affected by the number of molecules dissolved in it, the solute. Water is the solvent, the stuff dissolved in it is the solute. So quickly, we see red blood cells here. If we place them in distilled water, then water is a higher concentration out than in, more solutes inside. So we're going to have water moving inside. They'll swell and they'll burst and dump their their cytoplasm. If we place these cells in concentrated salt solution, then it's going to suck all the water out. Water is going to move from higher concentration inside to the lower water concentration outside, and it'll shrink the cell. We'll talk about that more and more. This graph shows the change in cell volume over time as we as we add a hyperosmotic solution. So this is a solution that is going to so we add a hyperosmotic solution here, and it is going to decrease the cell volume. But when we follow a weakly permeant solution to replace it, then the cell volume will increase, and there will be an increase in cell volume as this weakly permeant solute can go into the cell and counter the effect of the hyperosmotic solution. If there is an impermeant solute, then the cell volume will shrink and stay small. So if there is a solute present that can counter some of this, we can balance things out a little bit, or even push it to an increase in cell volume. So again, it's a very dynamic process of water movement that is not independent of solute movement. This is illustrated in this slide, where we have a general cell membrane here with a sodium pump that pumps the sodium out, and then there's a passive flux of sodium back in. So normally the sodium levels are maintained at equilibrium as ions passively enter the cell, and it is pumped back out. So there, if you add a metabolic inhibitor, a sodium transport inhibitor, then sodium will continue to go in, and the water will go in. That's what we saw in that previous slide. And so when the inhibitor blocks the active transport of sodium outward, the intracellular concentration of sodium rises, and water enters osmotically, increasing the cell volume, and then eventually the cell volume increases to the point where the cell will burst. So the movement of the water across the membrane, the osmosis, is a bulk flow, and it's the tendency of different substances. It's a bulk flow, so by definition, bulk flow is the tendency of different substances in a fluid to move together in the same direction due to a pressure gradient, as in the animal circulatory system. Osmosis, on the other hand, is the passive movement of water across a differentially permeable membrane in response to solute concentration gradients, pressure gradients, or both. So, for example, if a bag containing a sugar solution is placed in pure water, the water diffuse inward, higher to lower water concentration. So the diffusion of water across an ideal semipermeable membrane from an area of high water concentration to one of lower concentration. An ideal semipermeable membrane is one that is freely permeable to water but impermeable to solute. So we've already seen an exception to this general rule. There is the general rule for regulating water movement. The cell moves osmotically active solute, such as sodium chloride, first, then the water is allowed to follow, and this is an important example as in the regulation of water elimination by the kidney. So now if we go back to this illustration to look at osmosis, we have the little blue-white circles are now the water molecules, redefining them, and they can freely move through this membrane. And on the right-hand chamber, we have these large protein molecules contributing to the osmolarity of that molecule. Oncotic, the draw for the water now is from left to right. So the net flux is shown by this big blue arrow faintly in the back. It will be from left to right. So this illustration is showing something a bit different in a tube where now atmospheric pressure is going to be a factor. So there can be the hydrostatic pressure as well as the osmotic pressure. So water tends to move osmotically to regions of greater solute concentration from a hypotonic to hypertonic solutions. There is no net diffusion between isotonic solutions. We'll define these in just a moment. So here we have the membrane permeable to water but not to the solute, so which direction will water move? So we have a hypotonic solution over here and a hypertonic solution in compartment 2. So water is going to move from hypo to hyper, and we see what it does. It increases the fluid in compartment 2. Eventually this is going to have more weight, and it'll push to create a hydrostatic pressure that will have to be taken into account in all of these factors. So water will be redistributed at equilibrium. Well, now let's define tonicity and look at some examples again of the movement of water through an ideal membrane. So tonicity is defined as being relative to differences in osmolarity across a membrane, and it is the ability to change the shape of a cell, or in other words, the shape of a container based upon the water content. So here we've got 2 millimolar sucrose solution in a bag. We put it in 1 liter of distilled water. We get a net flux of water into the bag, swelling of the bag. Here we put it into 30 millimolar solution, higher outside than in, solute. Water concentration was higher in than out, so net flux of water is out. We get a shrinkage of the bag. Put it into a 2 millimolar solution, and we have no net flux. It is the net flux is 0, as much out as in. So this is isotonic. This is hypertonic outside the sucrose solution, so it draws water out. And this is hypotonic outside, so it's going to have water move in. So the red blood cells here, the hypotonic concentrations, the water diffuses into the red blood cell, which swell up. So relative to inside, outside is hypotonic. It has less solute, less tone to draw water, so the water is going to move in. Put those red blood cells into a hypertonic concentration, the water diffuses out of the cells and the cells will shrink. Isotonic, there should be no change, just as much water moving in as moving out. With plant cells, of course, we have the cell wall with the cytoplasmic membrane and the cytoplasm and the central vacuole. So if we put the plant cell in distilled water, hypotonic, then the central vacuole will have more water taken up into it or moving into it, and the cell stiffens but generally retains its shape because of the cell wall. So this produces turgor in plants so that they can move, change shape, or even stay standing. If, however, it goes into a hypertonic solution, the plant cell is placed in a concentrated salt solution, net flux of water is going to be out. The cell body shrinks and pulls away from the cell wall. So we see this here. This would be when it's in a hypotonic solution. The water moves inside the plant cell. The central vacuole squashes all the cytoplasm at the edge of the cell wall. In a hypertonic solution, central vacuole loses its water and the cytoplasmic membrane of the plant cell is pulled away from the cell wall. Well, let's jump then to a study across an epithelial layer with the mucosal membrane on one side and the serosal surface on the opposite side. So we saw this once before. So now, rather than just thinking of movement across one membrane, movement across a cell, we've got two membranes. So the cell decides where it's going to put certain proteins and what the permeability, the selective semi-permeability, will be of one side of the cell membrane versus the other side of the cell with its membrane there. So protein distribution is different. It's really quite amazing. So here we see the external mucosal side of that frog membrane, the skin, and the interior serosal side would be some connective tissue here. So the apex of the membrane of the cell and the base of the cell. So the apical membrane is has passive diffusion of sodium, and the basal cell membrane, or the serosal side, has a protein there, the sodium-potassium pump, that will pump that sodium from inside the cell to inside the skin or inside the body. And potassium that is taken in passively diffuses back into the interstitial fluid on the serosal side of this cell. So this active transport here sustains the concentration gradient of sodium, so it's low inside, high outside. So this passive sodium influx is occurring all the time, dependent upon the efficiency of this basal membrane with all these pumps in it. So we can have movement from chamber to chamber to chamber, depending upon the nature of the barrier in between them. So we got active solute transport from chamber 1 to chamber 2, and then a semi-permeable barrier that might just have passive transport with the other. And what we see is that the water follows osmotically with the active transport of a solute that's going to draw it in, and then barrier B, semi-permeable, might just allow the free movement otherwise. So a lot of variations on this scheme used by cells all over the body to accomplish their goals. For example, the kidney cell that we saw before with the tight junctions, we have the mucosal side and the interior serosal side with the interstitium. And we have active transport of a molecule that will freely diffuse through the lateral membrane. So the example that we just saw before. Active transport, water follows, and then a semi-permeable barrier that allows both of them to move. So we get active transport, diffuse out into the interstitial fluid between the cells, and then finally to chamber 3 through the basement membrane. So here's an example of what we just saw, and this does occur in the kidney as we take in sodium chloride, basically. The water comes with it. It's mostly just sodium. And then it freely passes through this and right in through here. And we reabsorb sodium and water. Now we can begin to think of bulk flow across the cell membrane. So we can have the formation of membrane vesicles to accomplish this. Exocytosis and endocytosis are the two examples. Exo, exiting, outside, from inside to outside. Endo, taking in, going inside, so from outside to inside. In this quick illustration of a cell, certainly not a human cell, for food intake is an example. So with exocytosis, it moves substances from inside to outside by enclosing them in a membrane packet, such as secretion. Endocytosis wraps the particle found outside the cell with the plasma membrane to form the vesicles to move the particles opposite to exocytosis. So endocytosis of a food particle on the right here, pinching off the membrane. We'll talk about that in more detail. To form a vesicle in the cytoplasm, it fuses with a secondary, with a lysosome to form a secondary lysosome. All these nasty things in here break it up. We absorb the food particles, the products of digestion that we need, and those that are the waste are released back into the environment through exocytosis. Not much of this going on in the human, but anyway, we see that as a prime example of endocytosis and exocytosis. Another way of thinking of endocytosis and exocytosis with respect to the number of vesicles inside the cell is that exocytosis, we end up with a decrease of the number of vesicles inside the cell. And with endocytosis, we increase the number of vesicles inside the cell. Of course, if these two dynamic processes are balanced, there's no net gain. But anyway, for simplicity's sake, we can think of it that way. Orient toward vesicle. It's a vesicle-mediated event. So when transport proteins in a cell membrane deal only with ions and small molecules, exocytosis and endocytosis, whereas exocytosis and endocytosis move larger packets of material across the plasma membrane. So for exocytosis, we see two examples: constitutive secretion, like maybe dumping collagen into the connective tissue of an area. So it's ongoing all the time. We used this earlier when we talked about cells. Or the regulated secretion, when a ligand binds to its receptor and we get a change in the cell so that we get a secretion of a secretory molecule. So again, exocytosis, we have a cytoplasmic vesicle that has to dock onto the plasma membrane, fuse with it, and then open up for that. So we lose cytoplasmic vesicles. Endocytosis, molecules, there's two forms, passive and active. Here we see passive. There's a concentration outside the cell of this stuff, and it just happens to get engulfed into this endocytotic vesicle being formed, this little pit that eventually is, there's a purse string effect here to pinch it off and bring the plasma membrane together. These are lipid bilayer phospholipids, so they'll just eventually fuse back together and we can pull it off. Cytoskeleton, obviously very much involved in these processes. Any change in shape or change in shape or intracellular movement depends upon the cytoskeleton of the cell. Now we can look at receptor-mediated endocytosis. So it's a membrane receptor often mediates that process. So we'll just go through the illustrations here on the left to the right. So initially we see some molecules, and these are lipoprotein molecules. You see them scattered around outside. They bind to lipoprotein receptors. The lipoprotein receptors, through the fluid mosaic model, are sequestered into an area. We see this illustrated on the bottom. Here are all the lipoprotein, the membrane receptors, and the lipoproteins are sticking to them. These proteins with cytoskeleton and the fluid mosaic model get all clustered into an area, and this triggers the binding of a protein, clathrin, onto this. Cytoskeleton pulls it in a little bit by connecting to the clathrin, so we start with a pit. So this is a clathrin-coated pit. On the inside of the membrane, a scanning electron micrograph shows that clathrin all over the inside of it. So we're looking from the inside at the surface of it here. A more three-dimensional view from the inside of the membrane. This is a two-dimensional view from a side view of the membrane itself. So you cut one of these and you look at it from the side. And this pit deepens. You see the clathrin particles on it to the point where now there's a purse string effect to pinch off its point of junction to the plasma membrane. And ultimately the clathrin associated with cytoskeleton pulls it away from there. And we have the clathrin-coated vesicle. Later that will lose the clathrin and undergo a variety of changes. We can now have even different aspects or specific examples of bulk flow: phagocytosis and penocytosis. So phago means to eat. It's a large-scale endocytosis, so endocytosis of large particles. This is the cell eating. Here we see the amoeba with its pseudopods, and they'll surround something and form a big giant endocytotic vesicle or phagocytic vesicle. Penocytosis is small-scale cell drinking, and it is endocytosis of fluids in smaller particles. This is very important in movement of molecules across the endothelial cells of some capillaries. So here we see a capillary that has different routes of movement. There can be water-filled pores through which molecules will move, lipid molecules, freely diffusible down their concentration gradient. Here we have the plasma proteins that are too big to fit through, so we have selection, size, exclusion, and then we have some other molecules that cannot make it through these pores, and they are not lipid soluble, they're water soluble, so something has to bind them on the inside membrane of this endothelial cell, and we get the clathrin-coated pit, the little teeny tiny one, pinocytotic vesicle, that shuttles across the cytoplasm to the opposite side and undergoes exocytosis over there to dump it into the interstitial fluid compartment. This is a bidirectional movement of these pinocytotic vesicles.